Matthias.
Yeah, thank you, Martina. Thanks to the FIDELIO organizers for the webinar. It's a real pleasure to be here. As you said, real pleasure for me always to talk about microRNAs. They're very close to my heart, so I take every opportunity I can to talk about that topic. I hope everyone can hear me well. My kids are not at home, so nobody should be streaming in the back, which should give me hopefully good bandwidth for the presentation. Okay, yeah, I'll start off. Okay, probably have to. Yeah, yeah, Martina mentioned that I'm affiliated with a company here in Vienna called Tamerna. And as companies so often like to have, we also filed some IP on, so intellectual property on some applications of microRNAs in the field of bone diseases. So just a bit of background. We, as Martina said, we founded in 2013. Here in Vienna, Austria, we moved the site from initially being a spin-off at Boku University, which is University of Applied Sciences and Natural Resources in Vienna. We moved site 2 years ago to the Vienna Biocenter, where we're now affiliated. We have a group of 12 people, all of those working on different R&D projects, essentially. You can see that this is a fairly recent group picture, as we're all nicely covered with masks, but I can assure you everyone is having a big smile, at least I hope so. So what we're doing, and what really since I started also my scientific career, I've been working on RNAs, specifically microRNAs. And then when we founded the company, it was on with the idea to utilize these RNAs and microRNAs, and the utilization or the application that we are after is as biomarkers. So essentially, biomarkers you can use obviously in medicine as molecular diagnostics to support decision making, you know, for doctors with their patients. And the second one we're looking at is also as drug development tools. So to provide decision support when you're developing a drug candidate during this process, you know, it's a multi-stage process, and especially it's there towards drug safety and drug efficacy. So that's what we are mainly developing. An overview of the pipeline of the projects that are currently running in the company. So this all started in 2013 with a project on bone diseases, specifically osteoporosis. That's also the most advanced project. We've passed through several stages and gates in the development, from discovery to validation to actually quite a large performance evaluation that's ongoing right now. This is on fracture risk assessment in post-menopausal osteoporosis. And then the pipeline spans also through other disease areas such as cardiovascular disease. I mentioned drug development and toxicology is one of the areas we're quite interested in, liver and metabolic diseases, and also very close to our heart, a project on aging and cellular senescence and utilizing microRNAs to make senescence measurable in blood, ideally. So outline for my webinar today is to introduce the group here and the FIDELIO students, first of all, to microRNAs, more of a basic introduction, so sort of to bring everybody up to speed, and then discuss applications of microRNAs on the basis of 2 case studies or 2 principles. Application either as drug targets in bone disease and as biomarkers, essentially for diagnostic use. So some facts about microRNAs. They are short, what the name already suggests, and by short, that means the mature form that you have at the end of the biogenesis pathway is around 22 nucleotides long, so that's actually fairly short, and they're non-coding. Non-coding implies that they're not translated into protein, but they develop their function, the biological function, as an RNA. As of now, and since the last update of the microRNA database that is called MIRBase, you can explore the database. As of the last update, which was 2018, we have around 2,650 microRNAs encoded in the human genome. So that's what we're working with generally. And in terms of function, they're regulators of gene expression, post-transcriptional regulators. That means that these fully processed short RNAs are incorporated in a protein complex. It's called the RISC, the RNA-induced silencing complex. And the microRNA actually functions as a guide for this protein complex. So this brings this protein complex to mRNAs, and that interaction at the end of the day results in an inactivation or repression, either of the translation or actually degradation of the mRNA. And there are certain estimations going on that about 70% of all human genes are in fact controlled by microRNAs. So how does this add up? That 2,600 microRNAs can regulate around 25,000 genes. So the system behind this interaction is actually only a short part of the microRNA sequence. Towards the 5' end of it is actually really relevant and required for the recognition of an mRNA target. You can see this nicely illustrated here. This is also called the C sequence. And so that enables actually microRNAs based on a relatively short stretch of hydrogen bonds that are formed with the target to not only find one specific mRNA or gene, but to potentially negatively regulate multiple different genes. This is not always a very strong inhibition, like 100% inhibition of translation or 100% repression, can also be moderate. But what has been found that individual microRNAs often control genes that also have some sort of coordinated action or fall into the same biological pathway. And by that, the individual microRNAs can be quite important and quite powerful in orchestrating really certain cellular programs required for cell function, but also deregulation of microRNAs could contribute to a deregulation resulting in a certain pathophysiological process. Another aspect of microRNAs that's quite useful also for studying them is that the sequences are highly conserved across species. So this is a simple blast alignment via the mere base of a microRNA. We are quite interested in microRNA 203A. And you can see that the sequence perfectly aligns, for instance, to the mouse, mus musculus, the human, or the rat ortholog of this RNA. And it also, not always, but often also applies to the function, so a conservation of actually the binding site. So we have here for this microRNA binding site, in effect, they're called RUNX2 or RUNX2. And you can see that the seed region required for the recognition by microRNA 203A in this gene is highly conserved between human down to, in this case, opossum. And that's, I think, also sort of illustrates probably the importance of these RNAs. So what about their transcription? Are those more like, some non-coding RNAs are considered as housekeeping RNAs or genes that are fairly constant and steady in their expression level. Well, for microRNAs, it's been known that they are highly dynamic in their transcription rates. And there is a nice paper that came out last year that utilized a method called SLAM-seq or metabolic sequencing. The basis behind this technology is that you add a compound, 4-thiouretine, that's incorporated into all newly transcribed RNA, resulting in a modification that can be utilized to make a base conversion, a T to C conversion. So by sequencing, then you can, and by looking for these conversions in RNA, you can actually know, or you can get an idea of what is newly transcribed, newly synthesized RNA compared to previously present, and you can really learn about the dynamics. And that has been shown that the biogenesis rates of microRNAs are really high, up to 228 molecules per minute. In this specific, it was a Drosophila cell line, but nevertheless, it really suggests that if a microRNA is turned on, it can be turned on quite heavily, and vice versa. So I think that's interesting to mention, but not also that the transcriptional regulation is highly dynamic. It's also that it's highly cell type or tissue type dependent. So that set of 2,600 microRNAs I've been mentioning is not turned on in all cells at more or less the same level, but this is highly cell type specific. And this is illustrated by the work here by Aaron Smith and colleagues. Was published a while ago, and taken from that paper is this heat map. This is only a subset of microRNAs, and you can see here in this heat map the expression level of these microRNAs across a range of different tissues, from muscle or heart tissue and others. And you can appreciate that you have several microRNAs where you only see a transcription so that light up, as I like to say, in specific tissue types, such as, for instance, this panel here that we refer to as myomeres, so muscle-specific microRNAs, or microRNA 122, which is a highly liver-specific microRNA. We have CNS, so central nervous system enriched microRNAs, and so on. And actually also for bone, bone was not in this study, but also for bone, we know that there are some enriched or specific microRNAs. So, of course, the next question is, in how far can this be relevant for disease state? To illustrate that microRNA regulation can be perturbed or altered in disease states. This is a recent study we were involved in together with the dermatology department in Vienna. And what we did here, we analyzed transcription of microRNAs in skin cancer samples. We utilized control skin tissue and then so-called perilesional to the cutaneous squamous cell carcinoma. That means in the surrounding tissue, and then directly from the tumor. So from the tumor lesion, we sequenced microRNAs, and you can see that sort of compared to control, several microRNAs are being turned on in tumor. This microRNA 1246 is, for instance, a really good example that was highly upregulated in the lesion, but not in the surrounding or healthy tissue. Thinking about this from a biomarker perspective, it's always important, you know, if you want to use an RNA as a readout of a pathologic state or a pathologic mechanism, this readout needs to be accessible. So we somehow need to be able to have material that we can really easily obtain from a patient without sort of having a huge safety risk for the patient. And by that I mean that we don't like to take biopsies, so cut out tissue if possible, but would like to take biofluids such as blood or urine or saliva for that purpose. So in terms of microRNAs, it's actually known quite well for a couple of years now that there are several mechanisms that facilitate or that are responsible for a controlled release of microRNAs from cells. This can happen through budding via exosomes or extracellular vesicles, or budding through directly from the membrane. And at the end of the day, these different processes end up in a release of, or result in a release of microRNAs and a stabilization, so that they're not accessible to RNases or enzymes that can degrade the RNA, but they're actually protected either by encapsulation in an extracellular vesicle or association to a protein complex. And so we have the ability to actually also quantify microRNAs in biofluids. We've ourselves been working with cell-free blood, serum and plasma, urine, saliva, synovial fluid, and CSF, but also milk and tear fluid and whatnot have been shown to contain microRNAs. In general, it is possible to actually also differentiate between vesicular exosomal RNA and RNA bound to protein complexes, or analyze both together. That's a possibility. And also nice part about this, if samples have been collected in the right manner and stored in deep frozen state, we can actually access biobank material or retrospective material that has been stored for more than 10 years. And so in case of this study, actually, then also where we've seen on a tissue level a high induction of this microRNA, actually also this resulted in a systemically increased level of this microRNA. So you can see that also in the serum of these CSCC patients compared to control patients, they showed an elevated level. And we obviously prefer to analyze this in serum rather than tissue. So to sum up this first part is that the main points being that microRNAs are post-transcriptional regulators of gene expression. Their transcription is highly regulated, it's highly dynamic, can be quite tissue specific. There are different mechanisms by which microRNAs can actively, through exosomes or protein complexes, be released from cells, or passively, of course, also if a cell dies, this can lead to a spillover or spill out of RNA. And those sequences are highly conserved across mammalian species. So coming to the second part, discussing potential applications, specifically in the context of bone. One study that I always like to show in this context is one published about 10 years ago, where the authors of that paper wanted to see what is the sort of the general or the overall effect if you delete or if you sort of down-regulate the post-transcriptional regulation of gene expression by microRNAs. So how can you globally achieve this? This can be achieved by knocking out an enzyme required for the maturation of most microRNAs, not all, but most, which is called dicer. It's an RNase required for the processing of a microRNA precursor into the mature form. So if you knock out dicer in osteoprogenitor cells in these animals, and you look at the using a collagen 1 Cre recombinase model, you can see you have a severe skeletal defect and severe impairment of bone development. So I think this really provided very good evidence for the fact that microRNAs are an important layer in the regulation of gene expression that's relevant to bone development as a whole. And a lot of groups, a lot of studies have taken this further in looking not only what's sort of the global effect of depleting microRNAs, but what's the individual roles of certain microRNAs in for instance bone turnover, so osteogenic differentiation, osteoclast differentiation. And there are several publications and reviews on this. I just for sort of to illustrate this, I just chose this figure from a review. That shows that by now we do understand that several microRNAs seem to fulfill key functions in regulating factors such as RUNX2, factors required for Wnt signaling, BMP signaling, etc. And the same also holds true for key factors required for osteoclast differentiation, such as the rank signaling pathway. I would like to focus on and then a gradual decrease again. So what this group here and what we did is we actually used mice, MIR146 knockout mice, so mice that don't have this microRNA and don't experience this upregulation. And a characterization of the bone phenotype in these mice was performed over time. And what you can see is that in those mice lacking microRNA 146A, they experienced a gradual increase in their trabecular bone volume per tissue volume. You can also nicely see this in these microstructure images, that those mice don't experience an age-related bone loss, but actually have an ever-increasing or steadily increasing bone density with age. So the question is whether this phenotype is more related to an osteoclast or osteoblast mechanisms. So individually it was studied what's going on with those two different cell types and what was observed in terms of osteoclast number. It was observed that the microRNA 146A knockout mice did not experience a lower number of osteoclasts. Only at month 12 there was a significant difference, but it didn't seem to be the driving force of this increased bone mass also at younger ages, that sort of there's a loss in resorption. So it was also shown that osteoclast activity between the knockout and the wild type cells was similar. So on the other side, what's going on with osteoblast in these microRNA 146A knockout animals, and it was found that osteoblast number is in fact increasing at an earlier time point already. Number of osteoblasts, but also number of osteoblasts per bone surface seem to be increased, and also resulting in a potentially higher osteoblast activity, such as shown by the uptake of the fluorocalcine in this case. So it was attributed that this sort of observation of an increased bone mass phenotype was attributed to an increased osteoblast function or activity. So then the next question that was asked is whether this also, or whether a reduction or a loss in microRNA 146A can actually also sort of rescue postmenopausal bone loss. And for sort of studying this, they used a model called the OVX, or ovarectomy model, where the ovaries are removed, resulting in an estrogen depletion, which again usually results in a fairly sudden decrease in bone mass. And so 3-month-old mice were randomized, both wild type and microRNA 146A knockout mice were randomized to either sham or overreectomy treatment and analyzed after 4 weeks. And what was found that the overreectomy treatment in the background of a mere 146A knockout mouse rescued the bone loss. So you can see here that there is still, after these 4 weeks of OVX in the knockout mice, they have maintained their bone and microstructure better than the wild type animals. Again, showing that also in this context, this microRNA could potentially contribute to bone loss. And this is just further illustrated on different aspects of trabecular bone volume, connectivity, and so on. But yeah, so the assumption was that actually this microRNA, or a loss in this microRNA, can protect or rescue postmenopausal bone loss. So that was the data from this study, and obviously the question is now, this is coming from a genetic model, a knockout model, that I think quite convincingly shows the relevance of certain of this microRNA for age-related or to age-related bone loss. Question is, are microRNAs generally druggable? Because obviously when we think about therapies, genetic models, or gene therapy is potentially an option now, but we'd like maybe to go the route via small molecules or chemical substances to drug our candidates, right? And so the question is, are microRNAs at all druggable? The answer is yes, and there's quite good evidence for this. So when we think about microRNAs, there are 2 regimens or drugging microRNAs that we can think of. One is that we actually would like to replace a microRNA. For this purpose, we can use so-called microRNA mimics. So these are synthetic oligonucleotides with certain chemical modifications that stabilize them, protect them from enzymatic degradation. There are different chemistries described that work quite well. And if we think about an approach where we would like to inactivate certain microRNAs, such as in the case of microRNA 146A, we'd like to inactivate this, there we can utilize antisense oligonucleotides, so complementary sequences. Or actually, and the challenge with administering RNA such as mimics or antisense oligonucleotides is the formulation. So we need to think of delivery systems such as liposomes. It's also now used in the case of mRNA COVID vaccines. They're also based on liposomes. Same can be used for microRNAs. But there's certain toxicity associated with this, or certain specificity to organs such as lung or liver. Quite hard to target bone with those. So also another option to inactivate microRNAs are small molecules. There's much more research going on now in this direction, where you can actually use conventional small molecule structure that usually have good solubility, good biodistribution, that target the secondary structures in these RNAs. And I think this is quite a promising new avenue to actually improve the druggability of microRNAs. To also illustrate this, also taken from that review, is just I'm not going into detail here, it's just a list of studies, different clinical studies, so in human studies up to phase 3 actually at the moment, where microRNAs or inhibitors of microRNAs are tested against their utility as new therapeutics. So to summarize this part is, again, initially I've shown that loss of post-transcriptional regulation of the gene expression by microRNAs impairs bone development. There's quite a lot of knowledge already about how several individual microRNA species, based on in vitro, in vivo assays, can regulate osteoblast-osteoclast formation. And specifically, this microRNA 146A, for instance, is a good example of an interesting drug candidate, as its transcription in bone increases with age. It seems to be causally linked to age-related bone loss. And as we have seen, loss of this microRNA can actually rescue these effects. Okay, coming to the third part. sort of what's closest to also our activities here at Tamerna, the utility is biomarkers. So as I said initially, when we think about biomarkers, we want to use biofluids, so-called liquid biopsies, and microRNAs can be detected in serum plasma and so-called circulating part of the circulating free RNA component. So something that we get out from this when you centrifuge blood after collection, the liquid part, the liquid layer on top of the cellular layer. So that's what we are interested, for instance, in working with. So obviously, always one important part when you talk about biomarkers is, how can you measure them, and can you measure them at all? And in general, for circulating free RNA and also microRNA, the two main methods that we use in our labs, also within the FIDELIO project, and probably the gold standard in this respect is RT-PCR. We require around between 10 to 200 microliter serum or plasma. For this, it's a rapid method, so turnaround, for the data is between hours and days. If you do larger panels, so analyze more microRNAs per sample, can take a bit longer. But it's a targeted assay, so you have to pre-specify which microRNAs or which RNAs you would like to quantify, and then design your primers and probes for that. So it's not that high throughput, or it doesn't scale that high in terms of the number of microRNAs that you can measure. So we usually go up, or have gone up in the past, to 380 microRNAs per sample. That's pretty much the maximum. On the other hand, it's an assay that's really gold standard and absolutely fit for being used in clinical routine. The counterpart to that, what we're now using more and more frequently, especially in an early stage of a project, is next generation sequencing, where the sensitivity is not as high. We can go down to 100 microliter of serum. It takes much longer to get the data, so turnaround, we're talking about weeks here, but the advantage is it's untargeted. And that means we don't have to pre-specify which microRNAs we want to assess in a specific sample. Basically, it's depending on your sequencing coverage and the amount of money you invest, you can detect as many as close to these 2,000 microRNAs that are there. So usually it's used in basic research or biomarker discovery. For me, always a very important aspect that's not frequently covered is, usually you would like, when you measure something, obviously you would like to measure variability based on the disease phenotype, or the pathology, or whatever you're interested in. So you want to see what does this specific treatment or condition have an effect on your RNA of interest. But you have to understand that this variation due to your model or your primary endpoint is in competition to other sources of variation in your data. And you have to be aware of that variation, and you should ideally really try to mitigate or control this variation as good as possible, so your signal to noise ratio for the variability you're interested in due to your disease phenotype is maximized. And when you're talking about microRNAs, we have several sources of variability, and one is pre-analytical, so that's everything that happens before you've frozen down your sample, and most frequently hemolysis and platelet activation during sample collection are the issue here. Then you have analytical variability, obviously coming from how well controlled your assay is, so on the RNA isolation, of the type of detection assay, the type of normalization you're doing. Also microRNA isoforms, I'm not going into the detail, can play a role here. And then you have biological variation. So on top of maybe the disease group that you're studying, there can be other factors that add variability on top of that. That can be certain circadian rhythm, for instance. So basically the daytime when you're collecting the sample. The time point in respect to the last meal, activity, smoking, and alcohol. So we know all of that affects microRNA levels, as it does the levels of many other proteins or other biomarkers. But those are all things that usually we should be able to at least document or control in a study. So we highly recommend actually of standardizing or controlling these factors. So just to illustrate hemolysis, I mean, to a certain extent, you can even by eye distinguish a non-hemolytic serum sample from a hemolytic serum sample based on the color. And that this does affect the levels of microRNAs is shown here. So even in this range, where, with an eyeball test, by looking at it, you might not be able to so clearly differentiate, especially in this area. You do see that this has quite a large impact on how many copies of these two microRNAs you find in these samples. So these are highly susceptible to interference by hemolysis. It doesn't apply to all microRNAs, but those microRNAs that are present in red blood cells. Platelets is another blood cell type that's full of microRNAs, so contamination with platelets can add variation to your data. And how can this happen? So the centrifugation speed during plasma collection is really influencing your platelet contamination. So the higher you centrifuge your plasma sample, the lower, and sort of the gold standard would be to use so-called platelet-poor plasma, double centrifuge plasma, where you don't have any residual platelet contamination. So just to illustrate this, when you compare single-time centrifuge to twice-lead or two-time centrifuge plasma, you can see that a platelet-enriched microRNA, you see a strong reduction in signal after 2 centrifugation steps. So the difference between these 2 signals is really the platelet contribution, while microRNAs that are liver-specific, so that are absent in platelets, are not affected by this. You see similar variation, either independent of whether plasma has been centrifuged once or twice. And I think that's also one important thing, is that it's hard to do any generalized statements on microRNAs. MicroRNAs behave like this, or are susceptible to hemolysis or platelet contamination. Each microRNA is really a biomarker on its own, with its own, interferences and dependencies on certain types of variabilities. I think that's important also to understand. So now, getting into a case study that we did here and that we published a while ago, and this was actually to compare microRNA profiles in the serum of type 2 diabetic patients and non-type 2 diabetic subjects with fractures and without fractures. So this was a really well-characterized cohort, but small cohort. So we have these non-diabetic controls and fracture patients, and we have type 2 diabetic subject without fracture and with a major osteoporotic fracture. The diabetic patients were quite nicely matched, except for the duration of diabetes, but for other factors, they were quite nicely matched. And as you can see, the fracture group, they had sustained quite a large number of even multiple fractures at some point. Really characterized, I would say, with a severe osteoporotic phenotype. So what we found when we compared around 380 microRNAs between fractured and non-fractured diabetic patients is what we found is that actually we found quite a large list of microRNAs that were regulated. And so the adjusted p-value here set to 0.05 for the cutoff. We had 23 microRNAs that were regulated. Only 1 was downregulated in fracture patients, while all others were significantly upregulated. So quite a strong effect. What was interesting to see is that when we compared overall this list of regulated genes to the list in fracture patients without type 2 diabetes, we saw that there's only a very small overlap, or actually there were hardly or much fewer changes in non-diabetic patients in response to a fracture. And there were only a few microRNAs that overlapped. And one of these microRNAs that overlapped was this microRNA 203A, which we've followed also a bit up. And what we knew at that time, then also based on literature, is that this microRNA, which is upregulated in serum of fracture patients with diabetes, actually does inhibit osteogenic differentiation via RUNX2 regulation, but also via regulating this transcription factor DLX5, and thereby impairing bone formation. There's a number of studies that have shown this, but also that this microRNA is actually responsive to BMP signaling, which inhibits this microRNA, or glucocorticoids. Dexamethasone actually increases this microRNA. So we came across this microRNA then in a later study on Sprague-Dawley rats, where we used again this overreectomy model. So we overrectomized part of these rats at 6 months, and then after 8 weeks of overrectomy, we randomized those to anti-resorptive treatment, anabolic treatment, or placebo treatment called vehicle, and we had a sham control in parallel. And then we let them sit for another 12 weeks, and at the end point here, at week 20, we analyzed a number of samples. So what we've observed at week 20, that the OVX animals with the placebo, so without treatment, had a strong reduction in PVTV. Anabolic treatment with teriparatide rescued this effect almost completely. Anti-resorptive treatment sort of resulted in a medium rescue. And this was also mirrored in osteocalcin, so in a bone turnover marker profile. But what interested us most was then, you know, what's happening in terms of microRNA regulation in these animals, specifically in bone tissue. And we've seen that in the bone of overrectumized animals, without treatment, we see a strong upregulation of, again, this microRNA 203A. This inhibitor of bone formation, sort of confirming our previous data from clinical studies. We also seen a slight rescue by anabolic treatment, but not anti-resorptive treatment of this microRNA. And this was mirrored sort of in an indirect profile of the target gene DLX5 in bones, also confirming what's been published before, that, you know, this microRNA most likely regulates this factor. What was nice to see then also, based on the serum samples from these animals, that we found a correlation between bone expression of this microRNA across these 4 groups and the blood levels. So again, showing that potentially we can use blood or serum as a readout or as sort of the matrix to analyze this microRNA and sort of being representative also of the changes that occur in bone tissue. That was a nice observation for us. And besides other microRNAs, we actually also observed that microRNA 203A shows a negative correlation to bone volume per tissue volume in this cohort. So the higher the levels of this microRNAs, the lower the BVTV ratio is, again confirming that this is an inhibitor or negatively associated to trabecular bone structure. Yeah, and you know, with this data, I'd like to sort of conclude that this is really some of the data that really got us very interested in microRNAs, to a certain extent microRNA 203A, but overall microRNAs in diabetic bone disease and bone disease in general. So very good. So the ITN FIDELIO project for Tamerna was a very good opportunity to get involved and continue doing research on these microRNAs. So I'm personally very happy to be able to closely work with 2 PhD students. So David, on the one hand, working here at Tamerna, who you know is looking at functions of microRNAs in diabetic bone disease, microRNA 203A, to which I just told you a bit of a history, why we're interested in that one, but also potentially other microRNAs. And then Ankita, who's in Sheffield, who's more on the clinical side, so aiming to measure and quantify microRNAs in blood samples obtained from diabetic patients with and without bone disease. So we'll see what the future data that we can generate in these projects will tell us about the importance of microRNAs for diabetic bone disease. I'm quite optimistic that we'll have some very interesting data in the next months. Yeah, and with that, I hope I didn't take up too much time. I'd like to acknowledge my team and collaborators specifically for the studies I've been showing you. Obviously our funding agencies, and also I'd like also always to sort of thank patients and animals, you know, that agree and more or less agree in terms of animals to participate and partake in these studies.